This is George Kobakhidze from Baruch, and this is for Bio 2010 summer session. So I'm going to talk about mitosis. And the goal of mitosis is to create a copy of a cell. Not something new, not something different, but an exact copy. So here's my cell. There's its nucleus. The nucleolus is this dense region on the inside. And we have DNA floating around in there. The goal of mitosis, again, is to simply make a copy. So at the end of mitosis, you can expect to have two copies. Looks like eyeballs here. So the goal is to make identical copy. Goal, make identical copy. Okay, and mitosis occurs in somatic cells. And this is opposed to another type of cell called germ cells. It makes more sense to explain germ cells first and then everything else. So your germ cells are your sex cells. They're also called the sex cell. And if you are a male, your sex cells are called sperm. If you are a female, your sex cells are called eggs. And then everything else in your body are somatic cells. So if it's not a sex cell, it is a somatic cell. So every other cell in your body. Every other cell in your body. So if you think of, muscle cells, hair cells, skin cells, liver cells, all those are examples of somatic cells. For germ cells, they undergo a slightly different process called meiosis, which we'll discuss in a future lab. Okay, but that's pretty much it for mitosis. Consists of two parts. The first part is called nuclear division. This is where the nucleus duplicates and then divides. So nuclear division. The second part is called cytoplasmic division. And you can see the word cytoplasm there because that's when the cytoplasm eventually splits off into two. Okay, now for chromosomes. Let's take a look at the nucleus of a somatic animal cell. So this is going to be an animal somatic nucleus. So here is just my nucleus, and we know that we have DNA here, but I'm going to draw it a bit differently than before. So let's say this is a piece of DNA. It has its homolog, and there are actually 23 of these guys. So 23 of these guys, and there are also 23 of these guys. If you put those together, this gives me 46 chromosomes. In this condensed form, we can call them chromosomes, but I'll mention something about that in a sec. I didn't draw all 23 on this side or the other 23 on this side because I don't have enough space, but know that they're there. For a somatic cell, we have two sets of DNA. This 23, is from one parent. This set is from dad. Instead of saying set, we will say we have one N from dad. And similarly, this set, these 23 chromosomes, this is from your second parent. This set is from mom. Or you can say 1N is from mom. Okay, so now if you take that, you have 1N from mom, you have 1N from dad, this gives me 2N. Another way to say this is diploid. So all somatic cells in your body are diploid. They have 2 copies of DNA, one from mom and one from dad. Now let me go back to germ cells for a sec. Okay. So as an aside, let's go back to our germ cells. Again, the male germ cell, the female germ cell is called the egg, and the male germ cell is called sperm. Now these aren't somatic cells, so they don't follow the same rules. They only have one set of DNA. As you can see, only one set of DNA. This again only has one set. When these two combine, we form the zygote. And now we have reestablished this diploidy because we need the instructions to create the entire body. So now we can say we reestablished diploidy. And if something has one set of DNA, we call this haploid. So these are examples of haploid cells. Everything else is diploid. Okay. I think that's all I wanted to mention for this. Okay, so now on to the cell cycle. The cell is mostly in 2 phases. One part is called interphase, and this is where the cell does whatever it's supposed to do. If it's a muscle cell, it's going to do whatever muscle cells do. If it's a liver cell, it's going to do whatever liver cells do. So under interphase, we have growth 1, we have synthesis, and then we have growth 2. The next part, this is when it's dividing. This is the mitotic part. This is a very short phase relative to interphase. This also has a few subdivisions. We have prophase, we have metaphase, anaphase, and telophase. And then under telophase, we have cytokinesis, where we eventually pinch the cytoplasm to make 2 cells. So again, in interphase, if you look on the graph here, this is where most of the cell is spending its time. So a majority of the cell's life is in interphase. Let's say cell's existence is in interphase. Okay. So during interphase, it's kept in its loose chromatin form, and this is so it can be available for transcription. So here is my, loose all over the place chromatin form. Now it's in this form, be able to make protein. But once it condenses to what I showed you before into these structures, we cannot make protein. It cannot read this. Cannot make protein. Okay, because the DNA won't be available. G1 stands for growth, and here we just simply, create organelles, because eventually we're going to need a copy of everything. And then the cell also increases in size, because again, in order to make a copy, we need to have the same thing. So when you chop it in half, you'll be left with 2 of the same product. Okay, now on to synthesis. This is where your DNA and your centrosomes double. So I haven't mentioned what centrosomes are yet. This is another organelle that is important for mitosis. Another organelle. Even though the DNA doubles, the ploidy number does not change. Okay, so let me show you an example. Okay, so a bit of terminology here. These structures, we can call them chromatids. You can call them chromatids. So now after synthesis, so after S, it's going to double. There's actually two of these little circles here called centromeres. So let me just say that centromere. These guys are called centromeres. The blue also doubled, and now we have two centromeres. Since they're on top of each other, it's kind of hard to tell, but it's there. Okay, so even though DNA doubled, ploidy number does not change. So if you look, this is still that one set from dad. This is still that one set from dad. This is also still the one set from mom. If you go to this picture, we still have only one set from mom, and this is only still that one set from dad. So 1n plus 1n gives me 2n, so we are still considered a diploid structure. The number of centromeres, however, doubled. Centromeres doubled, but you simply can't see it because they're on top of each other. Okay, so more on the naming. So now these two structures, we can call them sister chromatids to each other. So these two guys, they are sister chromatids. Or you can call this entire structure, so we can call this entire structure which consists of all that, we call this a chromosome. But yeah. Last thing to note, however, is a synthesis is an interphase, and your DNA does not look like this in interphase. So DNA does not look like this. During synthesis, during S phase, your DNA is in chromatin form, the loose configuration form. DNA is in chromatin form. We just draw it as chromosomes to illustrate the fact that doubling occurs, but your DNA is in the loose configuration during synthesis. Okay, so G2, just more quality control checkpoints, make sure everything is good for division, and then we can start with mitosis. And I will do this in the next video.